So lipid and sugar. Two important characteristics, two important components. One would be the sugar. O polysaccharides. The O just means oxygen. There's an oxygen linkage. So all of these here, those are your lipopolysaccharides. The blue things there are lipids, and the strands sticking up are your O polysaccharides. And again, oxygen linkage, although you can't really see it. Question?
Oh,
Are we
no.
okay? Okay.
So the lipid is,
Those are those little wall things that are sticking into the membrane.
and then the O polysaccharide is
Sticking
what's
out.
Sticking out.
Yeah.
Like little.
So the total thing is your lipopolysaccharide.
And the O is oxygen.
Just means oxygen linkage, that's all. So the O polysaccharides, they're important. Excuse me. They're important because that is where the antigenic specificity comes from for the gram-negative cells. Remember, no teichoic acid, but there has to be a mechanism for the immune system to identify the bacteria. So the O polysaccharides is how they do it. So teichoic acids do antigenic specificity for the gram-positive cells, the O polysaccharides for the gram-negatives. I'm sure the test question there's somewhere if I can just think of one.
Can you say that again?
She woke up. No, again, for antigenic specificity, teichoic acid is used for the gram-positives, O polysaccharides, for the gram-negatives.
Okay.
Again, there has to be some mechanism to identify the bacteria. Otherwise, no antibodies and then no us. The lipid part, those little balls that were in the membrane, they're called lipid A. I've never understood that, because if there's a lipid A, that implies there must be a lipid B. And there isn't. Only very agile. If there's a lipid A, you'd think there'd be a lipid B, and there isn't. So I don't know why it's called lipid A, but it is. Lipid A is the endotoxin. Endo means inside. In this case, inside the outer membrane. And toxin's a poison. The endotoxin is what makes us sick. When we're sick with a gram-negative infection, it's the endotoxin in the bacteria that's doing it. And as you can see, it's part of the membrane, the outer membrane. That's why endotoxin. Now this is, yeah,
You said that nutrients go inside the corns,
go through, yeah.
but why do they go inside? Is it bacteria? So
Well, bacteria need food just like all cells. I mean, they're living cells. They have to have a food supply. And not just food, but, building blocks of some sort. All living cells have to have supplies of lots of things. So there has to be some pathway of getting you through that cell wall. The cell wall will block many things, but it can't block everything. The cell would starve.
is that what we want?
What we want it, the cell doesn't want it, yeah. What we want is not the same as what the cell wants.
I'm trying to figure out why the CV plus the iodine will be able to come out of this one that has the outer membrane.
The membrane doesn't stop it.
Yeah.
The
is it
peptidoglycan.
permeability?
No, it doesn't. The membrane doesn't stop it. The question was, why doesn't the outer membrane stop in the gram stain crystal violet plus iodine? It's a larger molecule. The membrane doesn't seem to do it. That's all I can say. The peptidoglycan does, of course, but not the membrane.
Do both membranes get stained?
Mhm. They will. I know what I wanted to say. So the endotoxin, now gram-positive cells produce an exotoxin. Those toxins, exo means outside or outwards. Exotoxins are released from the cell. They're released from the cell. So again, the endotoxins stay part of the cell wall. The exotoxins are released from the cell. And they just go out into the plasma, into the blood. Now, for example, if we had a magic wand that could magically remove all the bacteria in your body. If you were sick with a gram-negative infection, and you took your magic wand, you waved the wand, all the bacteria would go away, you would no longer be sick because the endotoxin would be taken away with the bacteria. But if you were sick with a gram-positive infection, you waved your magic wand, the bacteria would go away, and you'd still be sick. Because the exotoxins are not part of the cell. They're all swimming around loose in your bloodstream, and you'll still be sick.
How do you kill them?
Pardon me?
I mean, how do you eradicate the exotoxin?
Well, you really don't. Your immune system would actually attack them, and they actually produce things that are called antitoxins. So it'd be a matter of time. I mean, you know, we all get better.
Right.
So the immune system does attack. But all I'm saying is the magic wand would prevent you from being sick with a gram-negative infection. It would not prevent you from being sick with a gram-positive infection. Yeah.
Well, what causes the release of the exo?
That, I don't know what causes the release. I can't answer that. It's just a property of what the gram-positives do. That's all that I can say. I don't know. The gram-positives release the toxin. The gram-negatives do not.
What does the toxin do?
It makes us sick.
Like,
But how do you distinguish, how do you distinguish between like the body's response?
Responses trying to get rid of the bacteria. But the actual being sick itself implies, for example, your metabolism slows down because many of the toxins destroy proteins. And in destroying proteins, they might destroy enzymes. Destroying enzymes, your metabolic level goes down. Or in destroying proteins, maybe it destroys building blocks. So you're not able to build structures that you normally would make. That can make you sick. That's the kind of stuff. It's interfering with your normal processes. You're right, fever, inflammation, as uncomfortable as they are, but those are really the body's responses to what's going on. But metabolic screw-ups is what the toxins do. I'm sorry, something. So the gram-negatives make an endotoxin, which is a lipid. The gram-positives make an exotoxin released from the cell, which is a protein. See, there are really a lot of differences between gram-negative and gram-positive cells.
I just have a question. Why is the exotoxin not in the book? It's talking about the endotoxin, but in the gram
It's got to be in there somewhere.
I just read it.
It's got to be. There'll be somewhere else. I'll see if I can find it for you. It's got to be in there.
Can you repeat you said that gram-negative is a protein?
No.
Or
Gram-negative makes endotoxins. Specifically, what's the endotoxin made from? Lipid A. That's the endotoxin. It's a lipid. Lipid A. And exotoxins from gram-positive are proteins. So obviously we like to kill bacterial cells. Usually we'll go after the cell wall. The advantage there is there won't be any side effects because our cells don't have cell walls. So usually we can go after bacterial cells without damaging ourselves. Lysozyme is something you'll be seeing quite a bit as we kind of move through. You've had A and P already, you've already seen it. It's a protease, an antibacterial protease. Breaks down proteins. Particularly effective against gram-positive. You would expect that. Proteases break down proteins. Gram-positive cells have proteins. Gram-negative cells don't. I mean, they do. It's just not very many. So you'd expect lysozyme to be effective against gram-positive and to not be very effective against gram-negative. So of course, it is going after the protein in the peptidoglycan, so those crosslinks. So when you do that, you are wiping out the cell wall. The plasma membrane, the cell membrane is still there. So you basically have a bacterial cell with no cell wall. And it's called a protoplasm. A protoplasm, no cell wall. So it's spherical because it just kind of swells up and everything. If you try that with a gram-negative cell, you would expect it wouldn't do as much damage. For the obvious reason, there's not as much protein. So there will be some damage, there is some protein, it's just not much. So there will be some outer membrane, some cell wall will still be there. You will get a spheroblast. That'll be a bacterial cell with some cell wall. With some cell wall. So it's also spherical, but not as spherical as for the gram-positive. So in other words, you are damaging some cell wall, not all of it.
When you add the
Lysozyme.
lysozyme to gram-negative.
That's what we're talking about, lysozyme. Now, if you really wanted to use lysozyme, I don't know why you would, but if you really wanted to, you could. You'd have to use EDTA with gram-negative. EDTA, ethylene diamine tetraacetic acid. You don't need to know that. I was just kind of showing off a little, but EDTA is something that you can use to weaken the ionic bonds, and then lysozyme can kind of go in and do stuff. But in honesty, I think it's a waste of time. So why bother? Osmotic lysis, that again is your osmotic pressure thing. More effective with gram-negatives than with human cells. I'm sorry, I said that wrong. I started thinking about it, it made no sense. So let me back up. Osmotic lysis is more effective with gram-negative than with the gram-positive cells. So I apologize, I messed that up. So that's actually something I already gave you. With osmotic pressure, gram-positive is the most resistant, the gram-negative less resistant, and our cells not resistant at all. Okay, so again, osmotic lysis then is more effective with gram-negative than with the gram-positive. And it's most effective with our cells. Our cells will rupture the easiest. And finally, for this big chapter, penicillin, again, I emphasize to you, antibiotics interfere with protein synthesis. So in the case of gram-positive, it would interfere with forming the crosslinks for peptidoglycan. So no crosslinks, no peptidoglycan. No peptidoglycan, no cell wall. No cell wall, no bacteria. Which is the whole point. But again, remember, antibiotics prevent new cells from forming. They do nothing to cells that are already there. That's up to your immune system to get rid of. But at least you won't be making any new cells. Gram-negative has less peptidoglycan, it's got the outer membrane. So you know already, the antibiotics are not effective. Again, maybe a little bit, but not much against the gram-negatives. Now, we do have medications. And I don't like this next part. They're called antibiotics. They shouldn't be. They shouldn't be. tetracycline and other stuff like that, they do operate against the gram-negative. They prevent the cell wall from forming. They inhibit the enzymes that assemble the cell wall. But there's a difference in how these two guys operate between the regular antibiotics and tetracycline. The regular antibiotics prevent the proteins from being made, so you're not getting the parts to make your cell wall. Tetracycline allows the parts to be made. You've got your protein, you've got your sugar, but you can't assemble them. See, with the regular antibiotics, with normal antibiotics, you don't have protein. So you're not getting a cell wall, it's because you're missing the parts. With the tetracycline, again, I don't like the term, but they do call them antibiotics. With tetracycline, you've got the parts, you just can't make them, you can't assemble them. So you're still not getting your cell wall. So they're both preventing cell walls from being made, but they're doing it a different mechanism.
So those still wouldn't be effective against the gram-negative.
I'm sorry.
Those still wouldn't be effective.
Well, tetracycline is, because again, it prevents a cell wall from forming. It's just doing it in a different manner than regular antibiotics.
Yeah, so like you still have all your symptoms that you mentioned on the all, but the tetracycline inhibits the ability of those
Enzymes to assemble. See, with the gram-negative, you've got your protein, you've got your sugars. You need to assemble them into a cell wall. Tetracycline prevents that. With your gram-positive antibiotics, you don't have proteins.
But how can you build this gram-negative
I'm
cell?
what?
How can you build this?
So the beginning of the quarter is what is fair game for your midterm.